Spores of bacteria. What are spores? Spores are the resistant inactive structures formed by some bacteria and fungi. They are formed by gram-positive bacteria. When their environmental becomes too harsh for survival, such as the loss of nutrients or food scarcity in their normal habitat. In such harsh environmental conditions, bacteria transform into a dormant state to form spores. Examples of spore-forming bacteria? Spore-forming bacteria are primarily gram-positive organisms that produce dormant, highly resistant endospores to survive harsh conditions, like heat, UV radiation, and disinfectants. Key examples include Bacillus species. Bacillus species are typically aerobes, oxygen-using, or facultative anaerobes. Bacillus spores are highly resilient, dormant endospores formed by Bacillus bacteria in response to nutrient scarcity. Capable of surviving extreme conditions, heat, radiation, chemicals, for years. For example, Bacillus anthracis, the bacteria that causes anthrax. Bacillus cereus, a common cause of food poisoning. Clostridium species. Clostridium species are strict anaerobes. Clostridium spores are often terminal, creating a swollen spindle appearance. For example, Clostridium botulinum, that causes botulism. Clostridium perfringens, that causes gas gangrene. Clostridium tetani, that causes tetanus. Clostridium difficile, that causes colitis. Why are bacterial spores called endospores? They are called endospores because intracellular origin. Unlike other spore types, like fungal spores or bacterial exospores, endospores are produced internally, starting with an asymmetric division within the mother cell. Internal development. The process involves forming a four-spored membrane structure inside the parent cell, which then develops protective coats before the parent cell eventually breaks down to release it. End of life cycle of spore. Germination. When environmental conditions become favorable again, the spore transforms back into an active, reproducing vegetative cell. What is the structure of bacterial spores? Bacterial spores, endospores, are complex, multi-layered structure, designed for protection, including a central core, cortex, and proteinaceous spore coats, allowing them to withstand extreme heat, chemicals, and radiation. Following are the structural components from inner to outer layer. Core. It is the innermost part containing the cytoplasm, DNA, a nucleid, ribosomes, and essential enzymes. It is highly dehydrated, only 10 to 25% water content, and contains high levels of calcium dipicolinate, which helps stabilize DNA and provides heat resistance. Inner membrane. It surrounds the core and acts as a permeability barrier against damaging chemicals. Germ cell wall. It is a layer of peptidoglycan located just outside the inner membrane, which becomes the cell wall of the bacterium upon germination. Cortex. It is a thick layer of specialized peptidoglycan that surrounds the inner membrane. It is essential for maintaining the dehydrated state of the core. Outer membrane. It is a lipid bilayer that separates the cortex from the coat. Spore coat. It is composed of multiple protein layers that provide resistance to chemicals, enzymes, and UV radiation. Exosporium. It is the outermost layer, present only in some species, composed of proteins, lipids, and carbohydrates. Key characteristics of bacterial spores. Dormancy. Bacterial spores show minimal metabolism and respiration. Resistance. They can withstand high temperatures up to 150 degrees Celsius and are not killed by boiling, requiring sterilization, for example, autoclaving at 121 degrees Celsius, to be eliminated. Structure. They consist of a core containing genetic material, surrounded by a spore wall, a thick cortex, peptidoglycan, and a multilayered protein coat that provides protection. Not reproductive. Unlike plant or fungal spores, bacterial endospores are not used for reproduction. One bacterium typically forms only one spore.